Today's video lecture is all about the coolest molecule that ever existed, the molecule that contains the secret of life, deoxyribonucleic acid, otherwise known as DNA. Okay, you guys as 14, 15-year-olds, freshmen, sophomores, you know a lot about DNA. But that is fairly new knowledge in terms of scientific research. So today's topic is all about how do we know what we know about DNA? How did we get there? What were the scientists that were responsible for this sort of research? So think about it this way. DNA, in terms of molecules, is actually a fairly simple molecule. Proteins, on the other hand, are very complicated molecules. So for many, many years, scientists were convinced that proteins were the carriers of genetic information. So the first couple of scientists that we're going to talk about are all about the scientists who determined that DNA was actually that all-important secret of life containing molecule. So the first scientist we're going to talk about today, and please make sure you're filling in your notes organizer, is Frederick Griffith. And Frederick Griffith was studying the types of bacteria that cause pneumonia. So this is all under number 1 on your notes organizer. So basically, he isolated 2 different strains of pneumonia bacteria from mice and grew them in his lab. So we're going to see sort of what his experiments were. Okay. Listen carefully to what he did and what he expected, because it's not what actually happened. So he started with a smooth strain of this bacteria, and when he injected it into a mouse, sad face, the mouse actually died. Then he had a rough strain of this bacteria, and when he injected that into the mouse actually lived. So he determined that the smooth strain was the strain that caused the disease. It caused pneumonia. And the rough strain must have been a non-disease causing strain of bacteria. So he asked himself, well, what happens if I heat kill that disease-causing bacteria? So he took that smooth strain, that disease-causing strain, and he killed it over heat. And when he injected it into the mouse actually lived. So he determined that he had killed the bacteria and they could no longer cause pneumonia. But then he said, well, what if I heat kill that disease-causing strain and I put it into a mouse injected with the non-disease-causing strain? So think about that. He has the non-disease-causing strain mixed with the heat-killed disease-causing strain, so what did he expect to see? He expected for the mouse to live. But what actually happened was that the mouse died. Okay, so that was very surprising to him, and he had to ask himself, why did that happen? What must have happened? So Griffith came up with this idea that there must be some sort of genetic information that can be transferred between living organisms. So he determined that bacteria could pass genetic information from one to another, and he called that transformation, that passing of genetic information from one organism to another. Okay, so that blank in 1B is genetic information, and here's your definition for transformation. So basically, the harmless strain transformed into the disease-causing strain because the genetic information was able to enter that heat-kill disease-causing strain of bacteria, making it once again disease-causing. Okay. Now, here's the problem. Scientists, despite the fact that Griffith said, you know what, I think it's DNA, they weren't convinced that it really was DNA that carried the secret of life. They were convinced that it had to be proteins because proteins were more complicated. So Oswald Avery came along and he said, you know what, I'm going to repeat Griffith's work in order to determine which molecule really was the carrier of genetic information. That's why he repeated Griffith's work. So he identified that the molecule that transformed the R strain of bacteria into the S strain was DNA. Okay, that was his conclusion, that it was DNA that was the carrier of genetic information, not proteins. So the rough strain of bacteria incorporated this DNA into their cells, changing them into that disease-causing strain. They transformed from non-disease-causing into the disease-causing strain. But again, scientists, they're stubborn. They really want to say, no, I just don't think that's right. I don't think it's DNA. I really think it's protein. So It wasn't until Alfred Hershey and Martha Chase came along where scientists couldn't disagree anymore, okay? Once, I'm going to go over this experiment. It's going to seem kind of complicated, but I promise you that it's really not. But just know that after Alfred Hershey and Martha Chase's experiment, it was 100% certain that DNA was the carrier of genetic information and not proteins. Okay, so Hershey and Chase studied viruses. And viruses are actually non-living particles, but they have genetic information. They have parts of living organisms in them, even though we still consider them non-living. And if you remember the characteristics of life, that should make sense why. They don't have all of the characteristics of life, but they do have genetic information. Okay, so they were studying viruses. They were studying a very specific type of virus called a bacteriophage. Okay, and a bacteriophage is a virus that infects bacteria. It's a very specific type of virus. Now, viruses, these types of viruses are made up of two parts. They're made up of DNA, and they're made up of a protein coat. That is very important when you talk about their experiments. So these viruses are made up of two parts, DNA and proteins. Okay, so what Hershey and Chase actually did was they were trying to figure out if they could tell what part of the virus entered the bacteria cell, then they would know what the genetic information carrier is. So they grew viruses in cultures containing radioactive isotopes. And think of these as sort of like glowing markers. Okay, so they had a glowing marker of radioactive phosphorus and they had a glowing marker of radioactive sulfur. And those radioactive isotopes attached to different parts of the virus. So the radioactive phosphorus attached to the DNA. The radioactive sulfur attached to the protein on the outside. So by looking at basically what the bacteria would then glow with, you could determine what was passed from the virus to the bacteria. So in the case of what actually happens in real life, when the virus attaches to the bacteria, it injects its nucleic acid, it injects its DNA. So this is going to now mark the bacteria as having the radioactive phosphorus. If it's attached to the protein coat on the outside, like the sulfur was, nothing gets sent into the bacteria. The protein coat stays on the outside. It doesn't carry genetic information. So the bacteria don't glow with the radioactive sulfur. So this allowed them to determine once and for all that viral DNA was injected into the cell and provided the genetic information needed to produce new viruses. So just a quick summary. They labeled the DNA with radioactive phosphorus. They labeled the protein with radioactive sulfur. They mixed the viruses and the bacteria together, let them hang out for a while, and what they saw was that the bacteria glowed with radioactive phosphorus, but they did not glow with radioactive sulfur, which meant that DNA was passed from the virus to the bacteria and not the protein. So Hershey and Chase were super important because they were finally able to conclude that it was DNA that was the carrier of genetic information and therefore passed from viruses to bacteria. Okay, and if you remember now we're going to talk about the scientists that we saw in the Secret of Life video. And if you remember, Chargaff was the German scientist who did not like Watson and Crick, but he accidentally helped them by telling him what his work was. And what he noticed was that no matter what type of living organism he studied, bacteria, yeast, herring, rat, human, he noticed that the amounts of A and T, adenine and thymine, were roughly equal to each other. And the amount of guanine and cytosine were always roughly equal to each other. Pretty close, almost exactly equal to each other, no matter what type of living organism he was studying. Okay, so look here at the rat, for example. You've got 21.4 and 21.5% guanine and cytosine. You've got 28.6 and 28.4 adenine and thymine. Look at the human there. Again, similar numbers. So he noticed that the percentages of guanine and cytosine are almost always equal, and he noticed that the percentages of adenine and thymine are almost always equal. So he came up with this rule, Chargaff's rule. C must always equal G, and T must always equal A. That allowed Watson and Crick to determine that C's and G's must pair together, and A's and T's must pair together. That's how they end up in equal amounts. Okay, he also noticed that there were two types of bases in DNA. So we have 4 nitrogen bases, and there are 2 categories of bases. There are pyrimidines and there are purines. The pyrimidines are the small bases. Remember, I put a small with a large, and oh, there we go. Now we have a pair. Okay, that's what Watson decided, or Watson figured out. So pyrimidines are the small bases, the single ring bases, and purines are the double ring bases. A purine always pairs with a pyrimidine. A pyrimidine always pairs with a purine. You don't have pyrimidines paired up with pyrimidines, and you don't have purines paired up with purines. You have to have a big and a small. Okay, now I'm going to give you a little trick on how to remember which bases are which. Pyrimidines, that word has a Y in it. Thymine and cytosine are pyrimidines. What do you know? They both have Y's in their names. Okay, pyrimidines are the bases that have Y's. Purines are the other ones, adenine and guanine. So adenine and thymine, guanine and cytosine. A purine always pairs with a pyrimidine. A large base always pairs with a smaller base. Okay, next came my favorite scientist of all time, and that was Rosalind Franklin, right? She was considered this sort of grumpy lady, and we know now that she was probably grumpy all the time because she was working in a highly male-dominated field, and she was this very competent, very brilliant geneticist, which back in the 1950s was not common for a woman. So she was using a technique called X-ray diffraction, which is basically bending X-rays around something and taking a picture of it. And in Rosalind Franklin's case, she was bending X-rays around a molecule of DNA and taking a picture. So you ended up with sort of like a shadow of the molecule itself. So this was her very, very famous Photo 51. This was that picture that Watson sort of, snuck a peek at and took off her desk without her knowing. And really what allowed him to put that final piece of the puzzle together to determine that DNA was a double helix. So this X shape in a Photo 51 taken by Rosalind Franklin indicated that DNA was a double spiral. Okay, last scientists to talk about were these jokers right here, Watson and Crick. Now these are the most famous of all because they were the first to put all the pieces of the puzzle together. They built the first accurate model of DNA, of the double helix. It conformed to all the research that was done by the other scientists. It was a double helix, which was seen in Photo 51. It had a backbone that was made of sugars and phosphates, and they alternated. It was sugar phosphate, sugar phosphate, sugar phosphate. The base pairs were attached to the sugar with A and T in equal amounts and C and G in equal amounts. So A's and T's were paired together and C's and G's were paired together. And like we saw in the video, when a scientist saw the model, they thought, there it is, that's it. They finally figured it out because when you saw it, it just looked right. And we know now today, with all the technology that we have, that Watson and Crick were absolutely correct. They built the first accurate model of DNA. And they won the Nobel Prize for it. Okay, so summary, here is the structure of DNA. DNA is often known as a twisted ladder because that's really what it looks like. It looks like a ladder that is spiraling around. So the rails of the ladder, meaning the backbone or the sides of the ladder, are represented by the alternating deoxyribose, which is the sugar, and the phosphate. So sugar, phosphate, sugar, phosphate, sugar, phosphate. That's what makes up the backbone or the rails of the ladder. The nitrogen base pairs are what make up the rungs of the ladder or the steps of the ladder. So cytosine and guanine, thymine and adenine, they make up the steps of the ladder. So nitrogen base pairs, alternating sugars and phosphates. Those are really the two major structures of a molecule of DNA. Okay, so Watson and Crick also noticed that hydrogen bonds can form only between certain base pairs, adenine and thymine, guanine and cytosine, and this is because of their structure. So we had a big molecule always paired with a small nitrogen base, a small nitrogen base paired with a big nitrogen base, a pyrimidine always paired with a purine. Okay, let me say that again. A pyrimidine always paired with a purine. And they also noticed that the different base pairs had different amounts of hydrogen bonds. So you can see in this image here, between guanine and cytosine, there were 3 hydrogen bonds. And adenine and thymine, there were 2 hydrogen bonds. And if you remember what we learned about hydrogen bonds back in, biochemistry, they're very weak interactions, which means they are easily broken, which is very important when we're going to talk about replication later on this week. Okay, so this is the base pair rules. Pyrimidines pair with purines. That is D3. 3. Pyrimidines pair with purines. Okay, so last on our list is we have the structure of a nucleotide. And a nucleotide you memorized is the monomer of the nucleic acid polymer. So it's this subunit that gets repeated all the time. A nucleotide consists of 3 items: a sugar, a phosphate, and a nitrogen base. Okay, the sugar, which is attached to the nitrogen base, and then the phosphate. All 3 of those items make up a nucleic acid nucleotide, the subunit that makes up nucleic acids.